Good evening, everyone. We're going to go ahead and get started. Thank you so much. Good evening, everybody. It's wonderful to see so many faces out there this evening, both familiar faces and brand new faces. Thank you for joining us this evening. I also want to acknowledge the more than 40 people who are joining us right now on Zoom. Thank you for tuning in. For those of you who don't know me, my name is Nikki Lewandowski, and I am the director here at the Institute of Arctic and Alpine Research, and the acronym that we use to describe ourselves is INSTAAR. We call ourselves INSTAARs. And tonight's event is the first in what's going to be a whole year, a long series of events in which we're celebrating our 75th anniversary as a research institute. We are the oldest and the coldest research institute at the University of Colorado, and we're very proud of that. Our institute was founded in 1951 by John Marr, a young ecologist who had a passion to understand all of the plants and animals that were hardy enough to survive extreme environments, such as those that occur up in the high alpine. And that theme will continue even tonight with Diane's talk, who will talk to us about some of those organisms surviving extreme environments in Antarctica. But our institute actually dates back further to the Mountain Laboratory, which was established in Tolland, just up to the west of Boulder in the foothills in 1908, and also connected with something called the University Camp. And in 1920, the Mountain Research Station was established, and that's sort of where we have our true origins, in the Mountain Research Station. And this Mountain Research Station is still there today, and we're going to have an event there later this year, a kind of big open house that you all will be welcome to attend. You worried about feedback? Yeah, I am too. Thank you. So INSTAAR was established and became very, very well known around the world for its research on alpine environments, Arctic environments, and also Antarctic environments. But today, we are an interdisciplinary institute where experts across all sorts of scientific fields collaborate on complex problems. And our mission is a mission to study the causes and consequences of rapid environmental change in cold regions and beyond those cold regions. Our planet is currently experiencing an unprecedented change in temperature at a rate never before seen on this planet. And so our work that we're doing today is even more important than it ever has been. And we're hoping to continue this work for several more decades, if not another 75 or 175 years into the future. And we're really glad you could be here to join us. Some of the events that we have planned for this year to celebrate our anniversary are up here on this slide, including our Monday noon seminars, which we've always done at this institute. We've invited some of the women who have helped shape INSTAAR into the wonderful institute that it is today to talk to us about their experiences and some of the research that they conducted when they were here, or some of them still are here and doing that wonderful research. And then on April 29th, we're going to have a celebration luncheon where we acknowledge all of our recent graduates, and we have some award winners amongst our students, and everyone is welcome to join that as well. And then on August 19th, we're going to have a big open house up at the Mountain Research Station on the Peak-to-Peak Highway just past Nederland. And we're going to have a lot of activities such as hikes to see permafrost, bird watching, you name it. And then at the end of the day, we're going to come together for a dinner. We're going to have an evening seminar from distinguished Professor Katie Suding. So please feel free to join us. We have a lot of other things we're cooking up, including at least one and perhaps two film screenings that you will be invited to, and maybe a discussion panel about greenhouse gases, and a fall seminar series where we're going to invite some of our newer faculty members to talk about the new research that they're working on. Without further ado, I'd like to invite Professor Mike Gusev up here to the mic. And I've asked Mike to introduce Diane because he knows her very well. Mike is a professor at INSTAAR and also in the Department of Civil, Environmental, and Architectural Engineering. He's also the Associate Dean of Research in the College of Engineering. Take it away, Mike.
Thank you, Nikki. Yeah, this is quite an honor. So I do indeed have all those titles, but before I had any of those, I got to be one of Diane's first PhD students, and I think that I feel pretty fortunate for that. And then I got the great fortune to continue to be a collaborator with her ever since. So I'm not always good at going off the cuff, so I wrote the words. So spent a lot of time on this, Diane. All right. So Diane came to see you and INSTAAR after a 17-year career at the U.S. Geological Survey. She has served as, let's see, I have slides, I forgot. All right, there we go. Those symbols on the left, or sorry, on your right, are what I want to talk about a little bit. So Diane was one of the co-PIs on the Niwot Ridge LTER. So that's at the Mountain Research Station right outside of Netherlands. And then she was also the executive, the chair of the executive board of the LTER network. LTER is long-term ecological research. And then she was a founding member of the McMurdo LTER. So that's the project in Antarctica and the Dry Valleys that she's going to talk about. And then served as the lead PI for about 6 years. When she came to see you, Diane brought a certain boldness of thought and creativity, as well as graceful tenacity, I'm calling it. She's an expert in dissolved organic matter and acid mine, acid rock drainage. And one of the most memorable experiments that I actually got to present on once, having nothing to do with this work, was to bring a milk tanker. We've all seen milk tankers driving around with an 18-wheeler. She brought a milk tanker full of Swanee River water from South Georgia, yeah, not local, to an acid rock stream in the mountains of Colorado. She wasn't trying to drain that swamp. Instead, she was seeking to determine how the organic matter transformed in the acidic stream and tease out basically the photochemistry of both the metals and the dissolved organic matter in the stream. And I don't know another scientist who would be so creative to dream this up and make it happen. It is one thing to dream these things up, it's another to make them happen. About the late 1980s, she started a new research career in Antarctica. She focused on the streams and lakes of the Dry Valleys, which she'll talk to us about tonight. And she became a leading expert on the freshwater diatoms in this ecosystem, studying how they respond to environmental stressors and changes. I say all this to note one thing. If you are one of Diane's graduate students and someone finds out, they go, oh, are you working on DOM, dissolved organic matter, acid mine drainage, or diatoms? That's what she's known the world for. I mean, worldwide. She is an expert in those fields. All right. As the lead PI of the McMurdo LTER project in Antarctica, she got to engage a lot of distinguished visitors. In this case, this picture on the right, she is talking to one of our program officers from NSF about some of her research. Now, I don't have time to go through an awful lot of things, but I will note that she has graduated more than 70 graduate students. And down here at the bottom, we'll jump ahead. Even when students were conducting 24-hour injections in streams, she baked a pie in a remote field camp in Antarctica to keep morale high. So, all right. Also, I am wearing this sweater, let's see, to honor Diane. When she was the lead PI of the McMurdo LTER, our PI group would watch The Big Lebowski many times in Antarctica. And we often thought about the many ways in which our challenges were somehow similar to those of the Dudes or Donnies or Walters. There were times when we got one disappointment or another, or in the parlance of the Dude, one gutter after another. And especially in Antarctica, it could be a bad weather day. It could be nihilists that broke the helicopter or the bowling ball that broke your piece of equipment, but we had to deal with whatever came to us. And whereas the Dude might have said, F it, let's go bowling, we decided to say, F it, let's go do some science. We found some alternative thing to do that didn't require the thing that got broken, or for whatever reason we had to pivot. But our PI group of 11 embraced the Dude's philosophy and kept moving forward, and Diane was our lead philosopher. Diane brought her bold ideas and tenacity to Antarctica. What you can barely see, these had to get reframed. Just down on the right-hand corner, those are weathered sandbags. Now, These are remnants of a 30-year experiment to divert glacial meltwater from one stream channel to another to determine how the other stream channel responded to being wetted for the first time in over 50 years. Again, there are few people who would ever dream of doing this, bringing hundreds of sandbags to Antarctica, building a wall with them, and even fewer who could pull it off, and then patiently and persistently study the responses for 30 years. But that's just my opinion, man. All right, so I'll end with with this, just to note that Diane has mentored a lot of great students to and through their accomplishments, graduated them, and then seen them go on to their own professional and personal successes. The first step towards that success was getting to work with Diane. And so to recognize her excellence and that she has earned a number of awards and honors, I list a few of those here. And then at INSTAAR and in every situation, she tends to lead by example with graceful and selfless tenacity to do the right thing for her students and her colleagues. In that vein, one of the roles that I omitted from this, I do feel badly, I apologize, is that she is one of the two co-editors of INSTAAR's peer-reviewed scientific journal, Arctic, Antarctic, and Alpine Research. So with that, I will finally hand this over to Diane. Please join me in welcoming CU Distinguished Professor Diane McKnight.
Oh, Mike, thank you. Thank you. Well, thank you very much for that introduction. And it is pretty much true that we did some of these fairly extreme experiments, and they're going to show up, not the one about the Swanee River water going to Colorado in this talk. So I am pleased to welcome you to the Dry Valleys. I have learned that many people are interested in Antarctica, and I'm hoping that you will see how special the Dry Valleys are. Many people are interested in penguins and seals. That's the Antarctic Peninsula, but I'm glad to assure you that there is one seal and one penguin in this talk. Videos. And so the first explorer to come to the Dry Valleys was Robert Falcon Scott. Many of you, or some of you, may have read about the heroic age and the biographies of these explorers. Robert Falcon Scott was not so impressed with this valley. He says it is worthy of record that we have seen no living thing, not even a moss or a lichen. It's certainly a valley of the dead. Even the great glacier, which once pushed through it, has withered away. This is a famous quote. We're going to be talking about some microbes that Robert Falcon Scott didn't see when he was there. I will also be talking about work done by Griffith Taylor. Taylor Valley is named after Griffith Taylor. He came about a decade later to the Dry Valleys. And I'll be talking about work that was done in the Nimrod expedition of Shackleton in 1907 or so. And again, these are the heroic age explorers, and we use their journals, their notes, to learn about how change has happened. So I want to emphasize that these are long-term studies in the way they start here in the journals of the explorers. I'd also like to point out McMurdo Station, which is where Robert Falcon Scott's base was, and we can see the Discovery Hut when we go to McMurdo Station. One of the themes of my talk will be not only long-term research and how that can give us understanding, but the role of teamwork. This is all a team effort, not only within our own research group, but the U.S. Antarctic program, the people who support us, the helicopter pilots. It's a team effort. And here is the notes of Griffith Taylor, me, and the three colleagues with whom he was hiking around the Dry Valleys. And then it's also another theme is being prepared, thinking about what might happen, how to be ready, and that's a team endeavor. And he says, ye complete explorer. So he is making notes about what he brought down his clothes, because when you're there, there's not much to do. In the case, if you don't have boots, you don't have boots. And then also, whereas Scott is known as the scientist who went to Antarctica and carried rocks back from the South Pole, Shackleton brought a marine ecologist, a biologist who studied the lakes. There's wonderful lakes. I'll be talking about them a little bit later. This is James Murray, and he collected these samples that went back to the British Museum, and a paper was published in 1911 about those diatoms. As Mike mentioned, we really like to study diatoms. As I mentioned, the explorers are fascinating to many people. I was very interested in the explorers when I was a kid. I became interested in the Antarctic in 5th grade. I was in a classroom with lots of kids, it was baby boom, sitting next to a cabinet full of biographies. And I just read biographies all day. I didn't pay much attention to anything. I'd been on double sessions the 2 years before. I just read biographies, and I read about Scott and Amundsen and the race to the pole, and did Peary really get to the North Pole? And I was afraid of falling into a crevasse, even though I was in suburban New Jersey. So, and when we published a children's book, which is highlighted here, which I got to present a book reading at the American Museum of Natural History, right under the blue whale. That was so cool. And that book, we needed to start also with the explorers. That was what our book editor said. Children will be interested in the explorers. So we have this same quote in that book. So I would like to tell you more about the dry valleys in terms of where they're located. Here's the map of Antarctica. This is the Ross Sea. Here's Taylor Valley right there. And so to get from McMurdo Station to Taylor Valley, we fly in helicopters, and often our gear is traversed across the sea ice and the Ross Sea to get to the Taylor Valley. So we being the scientists on the McMurdo LTR, we think of the dry valleys as being comprised of ecosystem components. And we have the glaciers, the soils, these vast expanses of very dry soil that is dry because the dry air comes off the polar plateau and carries no moisture. And then we have the lakes, these lakes in the valley floor. They have 4 meters of ice to get through. And then this feature here, which looks kind of rinky-dink compared to all these other landscape components, are the streams that drain the glaciers that provide the water to the lakes. And this is what I've mainly been studying. In addition, there are what we call cryoconite holes on the surface of the glaciers where there's biota and sediment accumulates and the biota grows there. And as you can see, to study this ecosystem as a whole requires expertise and knowledge in terms of different scientists with different backgrounds coming together. So this is a picture that includes some of our initial lead and co-PIs for the project in the early 1990s when we were getting started. I'm in the center wearing a helmet, so you can't hardly see me. But we like to point out in terms of the team, each season, and for several seasons, it's a team of postdoctoral scientists, research associates, collaborators, graduate students, sometimes undergraduates, working together. And we have a concept called the PI in the field. So the PI in the field is the lead scientist who's not just responsible for his or her students or postdoc, but for the whole team. So that's a key feature because as academics, we can't be there for 3 months, each of us. So as I said, McMurdo Dry Valleys is a cold place. We have Very cold temperatures, minus 47.7. It's also can be warm in terms of degree days above freezing, about 22 degree days, and it can be very windy. This value here says 51 mile per hour. It can be even windier than that, blow your tent away. And there's 5 months of darkness, but when there's sunshine, that warms the glaciers, and we get the stream flow. And I think this is a quote like from The Hobbit, either lots or none at all in terms of stream flow. So I have pictures of both to show you. The McMurdo Dry Valleys is a long-term ecological research site. Mike mentioned the Niwot LTR, which has been here at Colorado since 1980. It's one of the first LTR sites. So we have a network of 27 to 28. It varies over time. And this is a plot of average precipitation and average temperature. And we are the end members. We anchor that end member of being cold and dry. But this network allows us to evaluate and consider ecological concepts and get over the ecologists thinking of my backyard is like this, so everybody's backyard is like this. So we share ideas, and it's a very exciting environment to be in. So life is small in the dry valleys. There's life in the ice that I mentioned on the lake. Those are microorganisms. There's microbial mats on the streams that are freeze-dried all winter, and as soon as there's water, within 20 minutes, those microbial mats are photosynthesizing. We have the criconites that I mentioned, and then even in these very dry soils, there are cryptobiotic invertebrates. Nematodes, there's only 7 taxa of nematodes. The one you're looking at is named after Robert Falcon Scott. Scott named Lindsay. I don't know that he would appreciate this, but in any event. And then that is a tardigrade, a water bear at the bottom right here. And I have a video for you. I think this is the most charismatic of the invertebrates in the dry valleys. This tardigrade is eating some algae. This is what they live on. So about the microbial mats, it's not just one type of mat. I want you to pay attention. This is kind of a quiz. We call this the black mats. They're comprised of a type of cyanobacteria nostoc that is common throughout many streams, and it fixes nitrogen to grow. We call these orange mats. These are a type of mats that grow in the center of the stream. They don't fix nitrogen, but they're also a type of cyanobacteria that's common in Rocky Mountain streams all over. These are sort of pinkish. We call these red mats to distinguish them from these mats, and they are a little bit different. And then what do you think we call these mats? We call these green mats, and they are actually comprised not of cyanobacteria, but of eukaryotic organisms, chlorophytes, green algae. So we have these diverse mats, and we keep track of the biomass and abundance of these mats in the stream as part of the monitoring program. But what's truly charismatic in terms of charismatic microbes are these diatoms. And in the dry valleys, we have 51 different taxa of diatoms that we can tell apart. These are microbes that have siliceous shells that are preserved in sediments, but we can tell them apart. And these are electron micrographs of some of the common genera. So every little plug, any penny-sized plug of microbial mat will have 20 to 25 different taxa of diatoms in it. So that's one of our puzzles. Why are there only 7 nematodes in the whole expanse of the dry valleys and all these different diatoms? And for those of you who really like to look at pretty pictures of diatoms, we have a website which has like 150 different taxa from all around the Antarctic continent. It's the same genera of diatoms found in these polar desert oases, of which the McMurdo Dry Valleys is representative. And as I mentioned, the taxonomy of these diatoms goes all the way back to James Murray and Shackleton. The type collection for these diatoms is Caperoides. So I want to show you now a video of how do we collect these microbial mats. We put out orange flags and see where the mats are in a transect that we established back in 1994, and we collect the mats, we freeze dry them, we determine the chlorophyll, and we figure them out. And when I was working with Chad to upload this video, he called it gloves, because you can imagine it's pretty cold and your hands get cold when you do this work. Well, I want to get back to the beginning of the McMurdo LTR. Here's a picture of us. We look really jazzed to have joined this network that I was telling you about in 1993. And I want to point out some colleagues, Diana Wall, Paul Von Gera with the USGS, who was the hydrologist on the team, Bob Wharton, who was the lead PI, and Andrew Fountain, whom many of you know, a glaciologist. And one reason we looked so happy is our proposal we wrote in 1992 was awarded, whereas the first proposal we wrote in 1990 hadn't been awarded. In fact, one of the reviewers commented, well, this would be about as interesting as studying a pet rock. Anybody remember the pet rock fad? Do you remember that? They actually wrote it in a review. So we were pretty jazzed to get started on this. And we, each of us, came to this setting to this group from our own research background. So I want to tell you about how I got from studying dissolved organic matter, as Mike mentioned, to coming to the dry valleys. So my colleagues, and Dick Smith is here, we had this idea that we wanted to study the chemical nature of dissolved organic matter in water where there weren't any plants contributing to that dissolved organic matter. And we were with the Water Resources Division of the USGS. And this is an important problem because all of you drink water that has been treated to make it safe to drink. It's been chlorinated. And when the organic matter reacts with the chlorine, there's some production of chlorinated organic compounds which are regulated by the EPA because they can be tetragenic and carcinogenic if the concentrations are too high. And we wanted to go to a plant-free place to understand the dissolved organic matter there because it's confusing if you have different sources. Of course, our first proposal, as illustrated here, we proposed to go very ambitious. This has never happened. McMurdo Dry Valleys, Vestfold Hills, which is the Australian base, and Sydney Islands, where there are plants. I've still never been to the Antarctic Peninsula. And fortunately, that did get some interest in the reviewers, but the program officer, Dick Williams, when he was coming to INSTAAR to talk to scientists, said he needed to meet with us, and he explained that even though NSF is mandated to support the research interests of other agencies, NSF would not want to support us for more than 1 year, and that we would need to only go to the dry valleys. And so we wrote another proposal, right? We said, okay, we can do that. And we wrote a proposal, which was successful. Our other scientists who'd been working in the dry valleys were very helpful. They gave us advice on what kind of huts we should ask NSF to build for us and what to do and how to get along. And so in November 1987, we arrived. Here's our camp down here. This is Mount Falconer. And we were really excited. We discovered that the nickname the helicopter pilots had given our project was McNightmare because they had to shift all this gear to the dry valleys. And the program officer was now Dr. Polly Penhale, who was a great fan and supporter of the LTR over many years, but it was her first season. And she suggested to us, We might keep it on the down low that we were only there for a year since they just built us this big camp. Anyway, we needed a big camp because we realized we needed to measure everything that at the time. So we brought instruments for measuring dissolved organic carbon, for measuring gases, right, Dick? For measuring pH, temperature, pumps, gear. Here's our camp. And these are the columns where we isolated the organic matter from very large volumes of water, 500 gallons per sample. And as shown here, and we collected them in milk cans. And this is called a jiffy drill. George Aiken, who was part of this team, liked to say there's nothing jiffy about a jiffy drill drilling through 4 meters or 5 meters of ice. And you may wonder, how do we get those milk cans from the center of the lake to the camp? And the helicopters flew them in a sling load. We'll see a picture of a sling load. Here's a George Aiken carrying milk cans. INSTAAR has a fellowship in honor of George that we're very glad to have. So we had a very successful season. It was a thrill. And we came back to the USGS, and a couple months after we got back, a phone call came in from headquarters to the Arvada lab. They came and got me, and it was Bruce Molnia from headquarters saying, we need you to write an extension of your grant. And I explained, well, we got our samples, but that did not actually matter. So we needed to come back because we needed to come back. And so we got more samples that year, but I like this picture of Sarah Spalding and John Duff. We look very confident. We had this nailed, and we had another great season. And when we first arrived at McMurdo Station, we were told that our stuff was, or all these instruments and everything, the Halo text told us, well, there's a 2-week backlog, and you're not going to get there for a while because the traverse across the sea ice didn't go forward. And then we met with the program officer who explained to us that we needed to get all our instruments set up right away. And I said, well, and then it turned out, we later found out, well, we needed to get our instruments set up right away because this committee from the Senate was coming. And here was Al Gore. He had come to Antarctica to learn about the ice cores, to understand climate change. So this is back at his beginning of his interest, his deep interest, obviously, in climate change. And here I am, and there's John Duff, and what are we explaining? The importance of dissolved organic matter, which you just heard about. We didn't mention diatoms. So anyway, our next step was to write another proposal to come back and do what we call biogeochemical budgets. This is an underlying aspect of many of the long-term ecological research projects where we try to understand how nutrients and organic matter come together. And so this is the next team in 1990. And in order to do this, we needed to measure stream flow. And in the Water Resources Division, we're all about measuring stream flow. And we proposed to NSF to put in these stream gauges. And this map shows both the eventual names of these streams, but also the initial names at this era, where Green Creek, which is named after Bill Green, he had been in the Dry Valleys at Frixell and numbered the streams, F1, F2, F3, as you see. And this long stream, it was a cold period, wasn't flowing. So he named it, he didn't give it a number. And so when we came back, Paul Van Geer and I, to put stream gauges on these streams, Paul said, well, let's just call it F10. And even though it's between F8 and F9. And then later on, in the, when the LTR existed, my colleague, Diana Wall, when we were here at Frixell Camp, and I was explaining this numbering system, she said, "That's stupid." And she was right, because they couldn't remember exactly where we were going, and we had to name the streams, because people, if you're working together as a team and you're saying, "I'm going here, I'm going there," it's good to have a name. And so these streams are considered third order features in the Antarctic naming system. So we had to name them after people on a field team. We couldn't name them after Al Gore or even the helicopter squad. So that's just some background. So here's a picture of putting in these stream gauges. This is a partial flume, more sandbags to make a cutoff wall. And here is a picture of the completed gauge on Aiken Creek. George, we named this creek after George because it has the highest dissolved organic carbon concentration. He thought that was really cool. So it looks like it's broken. Here's the partial flume. We can know how much water is coming through that flume just by the depth of the water. And then when there's really high flow, though, it needs to be able to come over that cutoff wall and not knock it over. So it looks like it's broken, but it's not when there's low flow. Well, this season where we're putting in all these stream gauges, that was pretty exciting. But then something really exciting happened. This seal showed up, which is shown right here. This is a 6-foot-long teenage Waddell seal. Throughout the valleys, even Scott commented on the mummified seals. This is the first encounter with a live seal, which is dragging its body over the rocks and surviving by eating snow. In the Antarctic, there's the Antarctic Conservation Act, which prevents people who don't have permits from interacting with wildlife. So this seal was up above our camp that I showed you. So what happened next? The next thing that happened is the seal actually showed up in our camp. And the joke was it smelled the cheeseburgers. And so now we're living in backpacking tents. And there's this seal, and the helicopter pilots were there, and they called the senior U.S. representative, and they decided that the exception to the Antarctic Conservation Act applied to our field camp, and that this seal could be airlifted in a sling load, if you will, under a helicopter back to the sea ice at New Harbor. And you will see this happening in a minute or two. I felt that if there's ever an Antarctic exhibit at Epcot Center, they should make what you're going to see next the basis of a ride. It would be pretty cool to imagine yourself in a sling load under a helicopter. And this is going to come up in just a bit. So when I would be talking to children, my kids' school classes, and I'd be asked to talk to somebody else's kid's school class, and even eighth graders find this fascinating. So anyway, this is a story, and the seal was alive and well when it was dropped off and was trying to poke its head out of that netting. We don't know if the seal survived the winter, but it was doing well. And as I mentioned, I also have one penguin. The penguins are more mobile. This is a penguin that came to our camp. I have video of actually checking out one of the insides of a stream gauge. And now this penguin didn't need to be rescued via sling load. It's actually heading back to the McMurdo Sound and the sea ice on its own accord. It rescued itself, this is what we say, and you'll see pretty soon he dives into the moat ice, the open moat at Lake Frixell on his way back to New Harbor. And there he goes. But mostly it's microorganisms in the dry valleys. Here he goes. So in terms of how we work together as a team, we need to have a common idea of what the Dry Valleys is all about. And this is our graph that we use to visualize the Dry Valleys. And we use this to set up our monitoring stations to try to capture what's happening in these different ecosystems. And one of our basic resources is the continuous record on the Onyx River, which is the longest river in Antarctica, 32 kilometers long. And the record goes back to 1968 and was started by the New Zealand program. And we rely on that record. Here's a picture of the F6 hut where we measure stream flow. The next thing that happened in terms of major events in my Antarctic career is we published a paper about an Antarctic cooling trend in January 2002 in Nature, which is a big deal for scientists, and got a lot of press because it's controversial. That does this show climate change isn't real? But what's actually happening in the Dry Valleys at the time was that had been very warm and very sunny for 2 weeks. There was a lot of ice up there, and there was a lot of water coming down. And this is a picture of my colleague Diana Wall's ITEX chambers, her experiments. Wormherder Creek got this nickname from the helicopter pilots who thought they were herding worms into these little ITEX chambers. And the quote is by Barry Lyons, who was a PI at the time, "Diana, why did you put your experiment in the middle of a stream?" And I like to show this headlines from the Antarctic Sun-Times at the time. You notice here it says, "Ice cools as world warms." This is about our Nature paper. And then it says, "Heat wave melts ice, floods valleys." And the Christian Science Monitor reporter actually quoted the Antarctic Sun-Times to say it's a little bit more complicated than you might think. Well, eventually, Susan Solomon and her team figured out that some of this cooling was associated with the ozone hole, but then when this breaks through, we get these big floods. Here's the record of the Onyx River up to 2010, and here's this cooling trend, and here's the big event that I was just talking about. And during that time, the Onyx River had 700 cubic feet per second of water. That's a lot of water for any of you who know. So this idea, this event then informed our idea of how the Dry Valleys may be changing and what to look for in our long-term record. I am going to, well, since Mike's here, I'm going to talk about the importance of Mike's research in our research and understanding streams to help connect with how we inform research other places. I talked about how we could see in a plant-free setting the chemistry of the dissolved organic matter. In this plant-free setting, we can also see what's called the hypereic zone, the zone next to a stream that's under and adjacent to the stream. So when you go hiking in the mountains and you're walking along the side of a creek, you're walking on top of a hypereic zone. The water in the creek is actually moving in and out. But we can see this here, and it is a driver for how that sediment weathers and provides solutes to the lake and nutrients. So that can be, I have 3 degrees in engineering. I'm putting one equation up here for you. We can quantify that and then use that to interpret what's going on in other streams. So I'm going to skip this. And we did get back to our research on dissolved organic matter. When George and I went to the Dry Valleys, we collected about 7 or 8 grams of this organic matter, and we shared it with many of our colleagues who study dissolved organic matter and work on water quality. And they said, we need more. We've run out of sample. And so again, we wrote a proposal. This time my colleague Yo Chin, who's shown here, was the lead, and I, to go back to Pony Lake, the lake right out in front of Shackleton's hut, where the concentration of algae and organic matter is much higher. And what I'm going to do next is show you the movie that explains our research that we entered into the McMurdo Station Film Festival. And I warn you, well, Chad said we should warn the people on Zoom. It might be a little loud, but this is the context of this movie. You see it's very green. It's not that loud. Okay, you got the idea. But this is fluorescence spectroscopy and how it changed with that's more helicopters, algae. I have actually shown this video. It's at plenary talks, but it's all right. But the point is that this was a very successful, I'm going to stop it, very successful endeavor. And we told our program officer that this would last, the big sample, 68 grams instead of 7, 15 cubic meters of water. It would last 25 years. And it was gone in 15. So we may go back, or somebody will go back. But in any event, the next thing I want to talk about briefly is the importance of teamwork again. And I want to show this picture. Wormherder Creek was flowing here. It didn't have any water in it during this period, and then it flowed again. And we wanted to do experiment because we didn't know it was coming. You look at this graph. We don't know, even in December, if this would happen. And we all pulled together to do an experiment and make measurements. And this is my colleague, the soil scientist, Diana Wall, who recently passed away, who was a mentor for many of us. And what is she doing? She's collecting water from a mini piezometer, which is a very hydrologic thing to do. And we all worked together and studied this system that's so ephemeral. And here is a map showing how we made the pink, yellow, and blue lines are transects, where Diana and her team measured the invertebrates, try to understand why the ones that really tolerate water are there in the middle. And then the green, the orange are the orange mats that are there. Again, these orange mats have only seen water very rarely, 2 times in 20 years that we had been there. So just to give you a little bit of data, we found that there are only a few of these 50 diatoms that can really handle that very infrequent wetting up. Ludicola dolia shown here. We would see Ludicola dolia at 1 or 2% in the population all over the valleys, but then it went away. And now this is its home. It's, you know, the story about the bunny finds its home. This is the home for Ludicola dolia, these streams that flow very infrequently. So I'd like to move on to going back to Cape Royds, where we were able to go back and see how diatoms, how communities have changed since the time of Shackleton. And here is a map that James Murray drew. Was there GPS? No, but our historian colleague was able to figure out where these lakes were. This is Tyler Kohler, who was able to count these slides from the British Museum using the taxonomy that we had advanced to get an updated description and then figure out has change occurred. And the only place where change occurred was at Pony Lake. And just to give you a sense of how microbes can really tell you about change, this picture here is Ludicola mutacopsis, a particular type of diatom that was there at that time. And then this is a very long, skinny diatom, Craspedosaurus levissimus. And Lane, if you're here, you can correct how to pronounce that. Lane studied diatoms. anyway, just to show you, you can really see change in the biota. And we think that this change is driven by the lake rising, consumption of the silica, which is the basis of these shells. And so now the skinny diatoms are dominant. Well, I've been telling you about what we found out. I'd like to come to an end with what we don't know. Here's an updated Onyx River record. Here's the cold period, the big flood. And this is what's been happening since then. This is clearly driving lake level rise. This is a picture of the snout of the Taylor Glacier taken during Griffith Taylor's 1911. There's that picture, just to convince you how much water has come into Lake Bonny since then. This is that same spot now. So the lakes have been rising. This is a closed basin lake, but the microbes are fairly robust. And we see that these communities are persisting, but we still have some big questions. And one of the questions is, what is actually driving the generation of all this meltwater? I'd like to point out that this 2010 this is temperature on this axis, and this is how much stream flow came in from all those streams I was telling you about in the Dry Valleys, in Frixel Basin, different colors. And then this was a cold summer, and this is all the stream flow that came in. Some, lots, it was warm, and it was cold. What's going on? And we have, well, there's an unexplored hypothesis that this is also somehow related to the ozone hole. So I'm going to show you where the ozone hole was in December 2009 when it was warm, but there was no water. And that red star is where we are. The ozone hole intensity is a measure of the intensity of UV radiation. You may have heard of that. And so this is December 2009. It's fairly faint blue. If we go to December 2010, the ozone hole was much more intense and was right over the Dry Valleys in December, which is when it's warm. So I'm pleased to tell you that one of the INSTAAR graduate students, Joe McGuire, will you raise your hand? There he is. He's going to figure this out for his PhD. He just got back from the Dry Valleys a week ago, 2 weeks, 2.5 weeks. Every half week counts. So anyway, so we have lots to figure out. And that's one of the advantages or the benefits of long-term research. With that, I'd like to extend, again, great appreciation to all the people who have supported us, to you being here, to my husband, who's been awesome and supporting me for all these many seasons. And it's a pleasure to share the excitement of this research. Thank you.
We have a couple minutes for questions. Does anybody have a question? And if you do, you have to ask it into the microphone so the people on Zoom can hear you.
Thanks. I was wondering, how long ago do you think the McMurdo Valleys became dry?
When did they become dry? Well, there was a time when the glacier from McMurdo Sound advanced into the Dry Valleys, and then it retreated. I think that's about 25,000 years ago. And then there's periods when the lakes have been little brine pools, and it's been even drier than it is now, which was was about 1,000 years ago. So, and then if you go back millions of years, there's a lot of controversy, which we try not to get involved in. being in the here and now.
I was wondering, do you have to sterilize everything when you go in there so you don't contaminate these lakes?
Just to get to New Zealand, into New Zealand, you have to clean your boots, you do a lot of things, but we don't have to sterilize all our gear, and we are very careful going lake to lake, for example, but going stream to stream, we don't need to worry about that because the wind is blowing everything around. And we have found out that literally it's raining diatoms. There's a lot of microbes being deposited all the time.
Hi, Diane. Great talk. Oh, wow. It's a lot of feedback there. I was curious about your plot that you showed a bit earlier that showed the discharge from the different streams, but as a proportion, like with all the colors showing different proportions and like the contributions from different streams. And I was just curious if you were to plot that as like a just 100% contribution every year, are there like some streams that are more consistently contributing, and does that matter? I'm just curious about that.
Okay, well, that's a great question. I'm going to go back to a map of the streams. Let me see if I can find one. Skip that. So we have long streams and short streams. So the long streams, the meltwater is generated later because that glacier is higher. And you know, when you go out to the mountains here, it's colder. You go higher up, that's the adiabatic lapse rate. That is effect here in the dry valleys. So these streams that are draining these glaciers start flowing later because it takes longer to warm up. And then that water has to soak through all this sediment into the hyporheic zone before it gets to the lake. And there's some evaporation along the way. So whereas these short streams, the tongue of that glacier is lower elevation, they start melting sooner, they contribute more water. But during this warm period I was talking about, these long streams were contributing lots of water. So that whole system was really turned on. That's a very good question.
What's the most fun you've had?
What?
What's the most fun you've had?
What's the most fun? Well, every season has its high point. I would say that tracer experiment, so we do experiments where we add things in streams. Mike mentioned this. And I could have given a lecture that was just about all the cool tracer experiments we've done in the dry valleys, but that wouldn't have been as interesting. So I think the tracer experiment with the stream, with the worm herder team, and where we really pulled it all together, we had a great time, and we really learned something. That was a highlight, but there's many highlights. Yeah.
So we have a question online.
Do you do any research on springtails, columbola?
Not me, but yes, the team studies springtails. And springtails hide out under rocks, right? But they're elusive. Not every rock. But yes, so there is some research being done on springtails.
We are up against time.
Okay.
So I'm going to make sure we respect folks' time. Please join me in thanking Diane. Again, I want to say thank you so much for coming. And I also want to remind you that we have another half hour if you're interested in learning more about some of the student research that's going on here at INSTAAR. That's right out in the hallway behind the auditorium. We also still have some refreshments available. And again, we would love to have you join us for some of the future events that we're planning this year. So you can visit that QR code using your telephone, using your cell phone. I don't know why I said telephone, your cell phone, and it will direct you to all of the events, even the ones that we don't know are happening yet. So thanks so much for joining us tonight, and enjoy the rest of your evening.